molecular sieve. It's kind of like plates in a way. It's just, they have, it's just like basically they have a bunch of little wells. It has to try and fight its way through the aggregates. And so smaller fragments can just kind of meander their way through. The larger ones kind of get stuck. And you always want to run standard or a marker so that way you know what sizes you're looking at. And then if we were to do a southern blot, what would be the next step? So you run it on a gel. You run on a gel, you want to transfer it to a membrane so that you can then probe those DNA pieces. Okay. So an example of this being used was doing DNA profiling. Okay, so you have an outbreak of Staphylococcus infection, and you're having newborns are dying. So not good. Try and figure out, where is this coming from? Why are they all getting this? And so you kind of want to start testing different areas. You take swabs. You're testing the doorknobs. Is it just out there and so parents are doing it, or the nurses have it, does water have it? Is it in our natal unit? Where is this? And so by looking around, you're like, okay, this is what people who have it, this is what their pattern looks like. So you start trying to find, where does that match up? And they were actually able to find out that one of the nurses had it, who didn't even realize it, and was passing it on to the kids. And so this is one of those ways where you can go, okay, we found it, we can now isolate it, we can stop this. And so it's a DNA profile. So it's not a human DNA profile, but it's a microbial DNA profile. Does that make sense? Uses for, what would this, this would be RFLP, because you're doing restriction fragment polymorphism. Most likely you're just taking them, cutting it up, throw it on there. It's one of the great things with bacteria is it's small enough you can do that, genome-wise. Okay. Genomics, bioinformatics. So, genomics is the study and analysis of the nucleotide sequence of an organism, whereas bioinformatics is using the computer to do all of that, get all of the sequence data. And so, it is an amazing tool. There is so much on NCBI bioinformatics that you can use to find. And so down here we actually have a genetic code. So when they go through and do DNA sequencing, they use fluorescence again, because it makes it nice and pretty and easy to read. And you can actually read off the DNA sequence based on the color spikes. And so you can go through and look at all of these different, what are the nucleotides? What do they code? What does the proteins code for? And so bioinformatics is, once we started realizing how much information we were gathering, they needed a good place to store it, to house it, and to share it. And this is where NCBI is an amazing tool. So the National Center for Biotechnology Information. NCBI, it's a national resource. It stores, analyzes molecular biology, biochemistry, genetics. They keep changing their colors between blue and green, but the information stays the same. It's great because you can get a DNA sequence, throw it into BLAST, and be like, where did this come from? And so then all of a sudden you come up with different proteins, or different genes that make different proteins. You can look up, they have, PubMed has all of the scientific papers. You can look through the actual genes and genomes, figure out, where is this gene on the chromosome? Where is it located? What does it do? What is it responsible for? I make my biotech students, I give them a sequence. They have to go in and figure out what the protein is, so what gene it is. They have to figure out what disease does it cause, and figure out where it is in that chromosome. It's all things that you can easily pull off of NCBI. if you haven't checked out NCBI, you might want to, because it's amazing the amount of stuff you can find on there. Some examples you can have for looking at E. coli. You can look at the different structures, the different sizes. You can, you can look at the organism. Okay, we're looking at Escherichia coli. You can figure out, what is the semblance? It has one chromosome. How big is it? What is its GC content? So remember way back when we talked about GC content, it's one of the ways of differentiating microorganisms. You can see how many genes they have, how many proteins. I mean, E. coli is pretty well known and characterized, but for some of the other microbes, they're starting to get there. So they're working to sequence more and more stuff, and so you can find where things are related. How can genes pass from one to the other? You can really start to try and understand what's going on. Another cool little thing on CBI is they do structure summaries. So this is a cartoon of what the 3D structure looks like. So proteins have 4 levels of structure. So the first level being the amino acid sequence, second level being how they fold into either alpha helices or beta sheets, which is kind of they naturally fold. Third level is how they compact onto each other and maybe get those disulfide bonds, that ultimate 3D structure. And then quaternary structure is if you have more than one amino acid chains coming together to make the final 3D molecule. But they have all of these great little tools, so you can actually look at the proteins, you can rotate the proteins, you can also look at specific amino acids. So this is one of the things that I've done is I mutated proteins. I did a point mutant, and so I can look at it in here and look at where is it at, and you can turn the proteins around, look at it from every angle, and you're able to say, well, this amino acid is in this spot, which is supposed to be part of doing this, and how will it change having this mutation? So you can start doing some predictive things with this. But also it's great because you can see how it can help unfolding, like, how does that single amino acid change cause this protein to become inactive? So lots of great fun tools. If you do, Dr. Kubo teaches the bioinformatics, we're going to do a lot of this. It's also important because the microbial genome has, they're currently working to do the human biome, I think is what they're calling it. And so they're trying to genotype all of the microorganisms that are on a human. And it's somewhat disturbing how many that is, how many different species. But the more we know about the microbes, the better off we are, because we can maybe find new novel drugs or therapeutics or preventatives or pathways. Or we can also better understand them so that we can know how to kill them. So it's one of those things where the more we know about everything around us, the better. We're either going to take it as information, just for us to learn and preventative, therapeutic, or it's a pathogen. We want to know how to kill it without hurting us. They're also working on the energy generation. So how can we get this renewable energy? The idea was if you could go into a cornfield, harvest all of the corn, keep it for food, and then all the stouffer, which is the corn stalks and the husks and the leaves, if you could turn that into fuel, then you would be using the entire field, basically. So how can you take that cellulosic biomass, which has a lot of sugar, but it's not readily available, and break it down into sugar, which then can be converted into biofuels. So this is, big thing of how do we do this? Because getting through that cellulose is not really easy, the cellulose and the hemicellulose. So trying to just break that down into simple sugars, and then what are the best fermenting microbes to really turn that into biofuels. So this is all things, the more we know about the microbes, we're going through in CBI, being able to figure out what are different microorganisms, all different applications and benefits. So, agricultural applications. So you can produce transgenic organisms. So, this is recombinant plants and animals that are altered by the addition of genes from other organisms. So this is a lot of times you get the GMOs, herbicide tolerance. So they're able to take a gene from Salmonella, and it makes it so that they are resistant to Roundup. So Roundup ready plants. The idea being, if your crop is resistant to Roundup, you can just spray Roundup on the entire field and you kill everything that isn't your crop. Salt tolerance. This is a really big problem because as we water fields, if we go down to the layer, they end up bringing salt up, so we end up getting very heavily salted fields, or even just mineral deposits as you keep watering and the water evaporates and leaves all the minerals. So they were trying to come up with transgenic plants that can produce fruit and remove salt from the soil. So this was something that was actually done at UC Davis. Edward Blumwald was able to make tomato plants that have special vacuoles that will take up more salt. So the tomatoes can grow in a high salt soil. They'll take up the salt, they'll store it in their vacuoles, but the tomatoes don't taste salty. So then you can use the tomatoes for regular products, and if you remove the tomato plants from the field, you're removing salt from the field. So one other idea with this was to grow alfalfa. Because if you can make it so the alfalfa takes up the salt, then you feed it to cows and you no longer need a salt lick. So it's kind of a two for that one. They're also working at getting freeze resistance. So crops sprayed with genetically modified bacteria so they can handle some of these mild freezes. So ways that we can make it so we get better crops, or get more from the land. Pest resistance, so the Bt toxin. So Bt toxin is naturally producing. It's harmful only to insects. So organic farmers use this to reduce insect damage by spreading the bacteria on their fields. What they did for biotech was they actually inserted the Bt gene into corn, and they've done it to cotton and a number of other plants by now, but the idea was that then the actual crop is producing the toxins, so when an insect tries to eat it, it dies. And so you're able to stop or have a natural insecticide on the actual plant, or in the actual plant. More things for recombinant DNA is improving the nutritional value and yield. We're constantly trying to improve the yield of our plants. Trying to get tomatoes that have a longer shelf life, get them to ripen on the vine. There was this whole thing for flavor saver tomatoes. They actually had a much longer shelf life. It actually was available apparently in Davis for a while, but they were so much more expensive that it wasn't cost effective to grow them. But they lasted so much longer because they wouldn't rot as quickly. We talked about PGH, so the growth hormone, so you can produce meat with lower fat concentration, you can get more milk. Also, golden rice. Who's heard of golden rice? Never?
Golden?
Okay, so there was vitamin A deficiency was causing people in third world countries, they were going blind, and so the idea was, well, if we can make their food more nutritious, then we won't have some of these issues. So they actually found the gene from daffodils, I believe, for beta-carotene, so the vitamin A precursor, they put it into the rice. And so you can actually see the rice is golden colored. And so the idea was you're just eating rice, but you're getting more nutrients. And so they're still working on that one. They've already done that one, but they're trying to make other things. So the idea is one day maybe you can, instead of having to take a shot for a vaccine, you can eat an apple, or that's kind of like the dream, is to make it so that you can get all the veggies of broccoli and cauliflower in something that tastes good like strawberries. And so trying to make things more nutritious, more nutritional value. This also comes with dilemmas and ethics. There has not been a single study that's shown that there's a risk to humans or the environment. You'll have a lot of people going, oh my God, no GMOs. But a lot of times if you actually ask them what it is, they won't really know. And also, we've been doing this for years. there's nothing to say that a more natural method isn't better, because they were trying to make the perfect potato for potato chips, and so they kept crossbreeding, and they ended up accumulating excess toxins in the potato. And that's considered a natural method, but that doesn't mean it always has positive results. And so it's more of the, are there, rules and regulations to check? And right now with GMOs, those are more heavily watched than natural organics. So there's sometimes you're like, well, do I want the one that is being studied or the one where I don't know what they put in it? There's been a lot of standards that have been imposed on recombinant DNA technology, and with anything, it's not the techniques that are bad, it's how they're used. So you can create biological weapons with recombinant DNA. I mean, that's one of the fears, is what kind of things can you make? But just because it can be used to create biological weapons, should that mean we don't use it to create insulin? So it can become an ethical, sticky situation at times. Okay? And I have a worksheet for you guys.